Hello, this is the long promised short, I hope very short video on maternal effect mutations. And you can think of them as female sterile mutations because the offspring they produce don't survive, even if the offspring have normal genotypes. Okay, so with that, I will just go through just some thinking. And I want to use, we talked a lot in class about bicoid, and bicoid is there for probably the best example for us to go through now with a little more detail than just the way I talked about it at the beginning of the semester, because we have actual genotypes and things that we can talk about. So I want you to think about the bicoid gene, and I want you to think about where and when that gene is transcribed, and where and when the RNA is translated. Because when we're talking about a gene having an effect, a mutation having an effect, the mutation is going to have its effect where the RNA is translated and where the protein, if it's there or not, is. But I want to think about the whole process. So here's my really awful drawing of a ovarial. So this is an ovarial. These are the little egg chambers, the little squigglies up at the top here. These are the nerve cells, and the oocyte is the larger cell at the bottom. And where and when is bicoid transcribed? It's transcribed in nerve cells, and then it is, this is really not a very good drawing, sorry about that. And then it's transported from the nerve cells into the oocyte, and it is kind of glued, it's fixed to the inner membrane at the anterior end of the oocyte in all cases. Okay. So where it's transcribed in mom. It's transcribed in those nerve cells, and it's transcribed before fertilization has taken place. Because this egg hasn't even finished developing yet, but the gene has been transcribed. Okay. So, well, that's not good. Okay, but what happens if mom is mutant for bicoid? Then basically the RNA is not placed where it's supposed to be. Okay, so if we then look at this embryo, there'll be nanos, there'll be some hunchback RNA, and there'll be some caudal RNA, but there's not going to be any bicoid RNA at the anterior end of that embryo. And the embryo is going to die because no bicoid protein can be made because there isn't any RNA. And the embryo's genotype doesn't matter. So even if the embryo has a wild-type copy of bicoid, why does it still die? Because the gene is not being transcribed in the embryo. The embryo is completely dependent on whether mom put RNA in the anterior end of that egg or not. And even if it is wild-type, has a wild-type copy, it doesn't transcribe that gene itself, right? So yes, even if the embryo is okay, normal in terms of its genotype, because mom is sterile due to the inability to put bicoid RNA into the egg, the embryo is basically doomed, right? Think of it as mom not feeding the baby or something. If that, try to find some kind of analogy that will make it easier to understand. Okay, so what happens if mom is wild type for bicoid? Well, in that case, there is bicoid at the anterior end of those oocytes. The eggs are laid, they get fertilized, and now the embryo has the normal bicoid. This is actually a better pen than the one I used for the drawing. It's got nanos, it's going to have hunchback and caudal and so on, and the embryo is going to survive. Even if it's mutant for bicoid, yes, because even if it's mutant for bicoid, mom has already, before this egg was even fertilized, before we even knew what the genotype was, which allele it got from mom and dad, the bicoid is there, right? So she did provide for it what it needed for its early development. Now, when this embryo grows up, if it's a female, it's back in this it's back in this situation. So if if the embryo is a female, it's going to be sterile because it is mutant for bicoid, and now it's not able to put that RNA into the oocyte as it's supposed to do. Okay. But that's a quick. I hope this is helpful. I had quite a long conversation with two different students over the past week, and it was. I don't know if I completely understood what the confusion was, but I think if we look at it in terms of just a single gene and how that gene works, I think that's a better case than just trying to think, okay, maternal effect, you know, how does that, in just general terms, you know, and those crosses I showed with, you know, females that are this genotype and that genotype, maybe it was that was the confusing thing. And if we think about it specifically in terms of the bicoid gene or the nanos gene or caudal or any number of, there's lots more that mom has to put into the egg properly in order for the embryo to be properly patterned. And if she fails to do that, it doesn't matter if the embryo has a normal genotype or not. It's kind of screwed, right? Because she hasn't basically set up the pattern the way it's supposed to. So I kind of think something like packing a lunch or providing food or something is, you know, a baby can't provide its own food. So if you think about the embryo as like a baby, and the fact that even if when it grew up, it would be capable of making its own lunch, it can't do it unless mom provides it, you know, so the meals kind of have to be provided by the mother. And for me, that analogy seems to be for me, the clearest way. You may have to think of something a little bit different that might work better for you. Okay, I hope that helps. If not, please do let me know, because it's kind of, I think it's a fairly big concept, and so it's probably worth understanding how it works.